All right, so a couple questions on muscle contraction. So I'm going to play you this video first, all right? So you can get a little reminder of muscle contraction. The sliding filament theory of sliding filaments.
Muscle contraction is at the basis of all skeletal movements. Skeletal muscles are composed of muscle fibers, which in turn are made of repetitive functional units called sarcomeres. Each sarcomere contains many parallel overlapping thin, actin, and thick myosin filaments. The muscle contracts when these filaments slide past each other, resulting in a shortening of the sarcomere and thus the muscle. This is known as the sliding filament theory. Cross-bridge cycling forms the molecular basis for this sliding movement. Muscle contraction is initiated when muscle fibers are stimulated by a nerve impulse and calcium ions are released. The troponin units on the actin myofilaments are bound by calcium ions. The binding displaces tropomyosin along the myofilaments, which in turn exposes the myosin binding sites. At this stage, the head of each myosin unit is bound to an ADP and a phosphate molecule remaining from the previous muscular contraction. The myosin heads release these phosphates and bind to the actin myofilaments via the newly exposed myosin binding sites. The two myofilaments glide past one another, propelled by a head-first movement of the myosin units, powered by the chemical energy stored in their heads. As the units move, they release the ADP molecules bound to their heads. The gliding motion is halted when ATP molecules bind to the myosin heads, thus severing the bonds between myosin and actin. The ATP molecules are now decomposed into ADP and phosphate, with the energy released by this reaction stored in the myosin heads, ready to be used in the next cycle of movement. The myosin heads resume their starting positions along the actin myofilament and can now begin a new sequence of actin binding.
There we go. So what's happening right now, you see that phosphate is leaving the head? So when the phosphate is leaving the head, that's when this, the myosin head can go and attach to the binding site. Now you see that that's the dark purple pieces all along here. What exposes that binding site? Normally it's covered up with that band, that troponin. What opens up that binding site? You need some kind of ion to show up from a nerve impulse. It's an ion you've all been taught you need to take in your diet, right? Calcium. So calcium is released when the muscle signal sparks or gives that impulse. And that, you see these bands, these are the tropomyosin covered up, the calcium, open it up. Now, once the phosphate leaves and the head binds, then what happens? Let me play this.
The presence of further calcium ions will trigger a new...
So what happens right there? You see the muscle cell, the sarcomere, does this. What's this called? Contraction or movement, or it slides, power stroke, different ways to call it. But is the ADP on it anymore? No. So the ADP leaves, that's when you have the movement. Now there's nothing on there. But not for long, because what's going to soon go on to the myosin head? ATP. So pretty soon it's still stuck until the ATP binds to it. Then it gets released. You'll notice it was like this with the ATP. Now it bent to this, which is the power storing. This is holding energy. It's like, you guys saw a bow and arrow before? It's like pulling back that bow and arrow. It's holding energy right here. Now you have the ATP and the phosphate. And it's going to stay there unless you have the open site, and that's going to cause the phosphate to leave. It's going to bind, and then it's going to contract when the ADP leaves. It's going to keep doing that as long as what is present. The calcium. Calcium is opening up those binding sites. You always have a lot of ADP and ATP in your muscle. It's all dependent on that calcium being there. So it's just the cycle of ADP or ATP joining, pulling the head off, breaking up into ADP and phosphate, storing that energy, then attaching, ADP releasing, just over and over again as long as you have the calcium. Remember, yeah, so sarcomere, this from an earlier warm-up that we did. So those heads are along the actin filaments here, grabbing onto that central piece, central myosin. Sliding over and over again, sliding filament theory. So, as far as the warm-up, what causes the myosin head to store energy? Get stored, like pulling back that band on or the string on a bow and arrow. And what's the role of calcium? All right, so work on explaining those, please. Yeah, the answer's on in the diagram, so it's there. Really good hints. There are some good hints, Oh, make a guess, Bree. Hey, Bree. headphones out. Why don't you put your phone away for a little bit, okay? Like, try 5 minutes, okay? And then maybe look, and then maybe go 10 minutes. Try to gradual pull away from that. What's that? The answer's up there. How's your pickles video coming, Valeria? The pickles video. Pickles or no pickles? Can you have it by Thursday? Anybody, you already started it. Okay, we'll start. Use the people you already have. Valeria! Well, let's look, Valeria, because there's good hints. Myosin head is right here. What do you see on that myosin head, Valeria? You see ADP and what? PI. What's PI? What does that P stand for? Anybody? Phosphate. So, Valeria has ADP and phosphate. Is this in the energy storage position? When it's like that, is that storing energy? It is. Okay, this is the energy storage position. It says relaxed state, but that's referring to the muscle itself. The myosin head here is storing energy. It's ready to go. That's why I say it's cocked, kind of like, this is like a gun reference, like a hammer on a gun cocked back. You know, God, why do you always bring guns into stuff? You know, it's culture. Anyway, so that's why I use the bow and arrow. It's not quite as a little better, I think. Or a slingshot. You guys know a slingshot, right? It's like holding it back. Ooh, that'd be good. We'll demo with some slingshots. So this is storing energy. There's an ADP and a separate phosphate molecule. But what was that before it turned into those separate ones, Valeria? What was it? What is ADP before it becomes ADP? It rhymes with ADP. ATP. All right. So, in order to store energy in the myosin head, ATP attaches to the head and then breaks into two separate molecules, phosphate and ADP. And that stores energy on the myosin head. That separation of the ATP and the ADP and phosphate. Okay, so ATP ADP and phosphate. And that's shown right here on the head, the ADP and phosphate. So that's storing energy right there. It's ready to go. It's ready to snap back once it gets ready. What causes it to snap back once it attaches? Phosphate leaves for it to attach, and then the ADP leaves, and that allows it to expend its energy, to like let go of the slingshot. Rubber band. That's releasing the ADP. And then it snaps back. Okay? What does calcium have to do with all this? Marcos? It's an opening for what? Okay, it creates the opening here on the binding site so the myosin head can attach. So it moves the tropomyosin out of the way so the head can attach. Try my best handwriting. Normally the binding site is blocked by the tropomyosin. So the calcium shifts it over like we saw the deep purple little circles in the video previously. So the myosin head can then attach. As long as there's calcium there, what's that muscle going to do? It's going to be contracting. What do you need to have the calcium there? What needs to be present? Or what needs to happen at the muscle cell? We need to have, has to do with that microscope right here. Medulla oblongata, brainstem. What is this? What's this all about? What kind of cell? Brain cell or nerve cells, neurons, right? So there needs to be a nerve signal at the muscle. Then it'll release the calcium, then it'll contract. So calcium is really important for muscle contraction, all right? If you have calcium issues, you're going to have, what's the one muscle you always need to have going that can be sensitive to calcium that if you don't have it, you're going to die? Your cortisone, see, your heart muscle, okay? So you always have that calcium there, okay? So that's why people having heart trouble, that's one of the things they'll be looking at, is your calcium levels, okay? Making sure that's the right level so your heart can function. Okay, muscle cell. So it's complicated, right? A lot of parts involved, sliding filament theory. And that's probably, of all the tissues that we have, the muscle tissue is some of the hardest to actually see some structures. Really the only thing, you're not seeing these sliding filaments at all. Probably the only detail you really see is in the striated muscle. You see some little bands, which are in between the sarcomeres. So the rest of them have a lot more detail than the muscle cells, unfortunately. Our holiday party is going to be the 15th. That's Friday. Okay, we had a good trial run with our Chick-fil-A, but we need to generate a flyer, and we need to do some finalizing of our budget. So this meeting Friday, we really want to get that figured out, get how much we're ordering, what we're going to order, whether we're going to go for sandwiches and nuggets, or just do one or the other, or a combination, and so forth. So Friday is important. We want to get that budget submitted so we can buy stuff for that. It would also be fun to make a holiday tree. Okay, maybe a fireplace. Because we'll be in here, So it'll be easier to do the decorations and stuff that way too. Okay. So that's on board. Thursday, we'll have a lab practical. What I'm going to do is set you up, and you're going to identify the different tissues. Not all of them. I'm going to select some different tissues, have you focus them, look at them, and tell me what those tissues are. Okay, so muscle, nerve tissue. Is it liver tissue? Is it skin? So as you go through finishing up today your tissues lab, make sure, you could identify the different tissues, okay, if you see them, all right? Maybe taking pictures of what you can, right, will help you with studying and look at your diagrams. I also want to come through, 5 of you have turned in or 6 of you turned in your problem questions for your IA. So I need everybody to turn in, that's the 3rd assignment, because I really need to know what you're going to do. Okay, we need to know that. So we need to get you your stuff and get the ball rolling. I'm also we'll do t-shirts too. So if you have your $5, give me that, and then we'll get you your t-shirt. Okay? All right. So go ahead and get started with finishing up your tissue experiment. Okay, so remember, structure and function, make sure you nail that down. If you have a question, let me know. Okay? What's that? You want to do as many different stations as you can. If you did not finish your last tissue sample, then finish up where you left off at. Oh, by the way, so remember all the different ways to adjust the lighting, but also some people had a lot of success with, there's one of those eyepieces that you can adjust, to get a better view. A lot of people had trouble until they adjusted that left eyepiece to really bring both in tune. So I really want you to focus through both. Get those eyepieces adjusted so you can look through both of them. It's a lot less strain on your eyes. Okay? All right, any questions? So go to whatever microscope you were at if you need to finish it, or shift to a different one. Then you can move to a different one.